Dorothy Buck and DeWitt Saunders, who are going to tell us about DNA topology, where maths meets molecular biology, essentially the subject of this whole meeting today. Dorothy is reader in mathematics at Imperial College, and as you see, she's also at the Department of Mathematics and Center for Synthetic and Systems Biology at Imperial College. And she is one who has successfully made this great leap from pure mathematics to molecular biology and has made very dramatic contributions in that area. DeWitt Saunders, who will speak immediately following Dorothy from Florida State University, is one of the pioneers in this area of topological ideas and concepts applied to DNA, the dynamical processes, enzyme action in DNA, and so on. So, Dorothy, may I leave the floor to you?
Thank you very much, Keith. Can you all hear me okay with the mic? Sorry. It's in an awkward place because I don't have a belt. So I'd just like to open today by giving you all an introduction to the title of today's Open for Business, which is DNA topology. So the first 5 minutes or so will be to remind you of some interesting features of the familiar DNA molecule. And the second bit will be on the second word in the title, topology. So for those of you that aren't already familiar with the second word, don't worry, we'll chat about it later. So it's, of course, very fitting that we talk about DNA topology here in Cambridge, since it was in the Cavendish labs that Francis Crick and Jim Watson, based on crystal structures from Maurice Wilkins' lab done by Rosalind Franklin, discovered the double helix structure of DNA, which is, I'm sure you all know, is that of a twisted ladder. And the fact that it's not a straight ladder, but a twisted ladder around its central axis is going to be important for us later. So this is the iconic photograph of them looking at it. But I want us actually to think about a photo of the two of them taken several years later with a little rendition. So this is Crick and Watson still. And this is our DNA molecule. And what you see in this picture a little more clearly than the other is the axis of this twisted ladder. So these are the alternating sugar phosphate backbones. Here's one. Here's the other. But supporting all of that in this molecular model is an imaginary axis. And it's going to be that axis that we're going to concentrate a lot of our attention on today. And what you've seen in this ray tracing here is that actually, although in this photograph, this axis is linear, that it could also, for example, be circular. And so here's a picture of this circular axis, imaginary axis of our real DNA molecule. And this axis can often be circular in bacterial genomic DNA, in our own mitochondrial DNA, in chloroplast DNA, many situations. And of course, in the lab, plasmids worked on in many molecular biology labs, including mild ones, then the DNA molecules are circular. So not only are they circular, so this is an electron microscopy image of a circular DNA molecule. So the DNA is coated with a protein, REC-A, to thicken it and stiffen it, and then it's visualized using electron microscopy. Here's a picture of a circular DNA molecule, and up here in the second picture is another circular DNA molecule. But you see that this one doesn't smear out in a nice, open, flat way. And we say that that kind of situation is when the DNA axis has what we call rive. And we can quantify that rive. There's a precise integral formulation, partly due to Gauss for that rive, but for now, we can just think of it as the overall projections, the average, of the signed number of crossings of the axis with itself. So this rive is what we refer to it mathematically. Biologically, this idea of rive is often referred to as supercoils. Since if you have a helix, generously supplied to us in everyone's office by the Newton Institute, unfortunately left-handed, if you look at the axis of this pseudo-DNA molecule, and you begin to introduce torsional strain into it because you're chatting on your phone, then you start to generate supercoils. So that's coils. It's a helix already. It's a coil. But now you generate coils of coils, these supercoils that show up on your phone. And so this supercoiling, or rive, plays an important part in DNA, as we'll discuss later. So remember in the very first picture I showed you that the DNA molecule is actually a twisted ladder. And there's an important correlation between rive, this quantity of where the axis itself is contorted in space, and the property that the sides of our ladder are twisted around the central axis. And so this is a picture, in case you can't see up close, of looking at the interplay between twist and writhe. So here's our small segment of DNA here. In this first picture on the left, the axis of the DNA molecule is very highly contorted in space. If you looked at it over all projections, from points on a sphere, you would see many potential crossings. And yet there's very little twisting of that tubing around itself. Yet if you pull it out very tightly, very straight, you get very little writhe. And if you can see on my phone cord or up here, we get more wrapping of the edges of the ladder, the sides of the ladder, around this central axis. So there's this interplay between, sorry, this has some kind of memory, but in an ideal world it doesn't. There's an interplay between the twist and the writhe of this twisted ladder, of this DNA molecule. So twist and writhe are two interesting mathematical, and twist can also be defined mathematically, properties of these DNA molecules, but we're going to consider I'm biased, but an even more interesting property, which is the fact that the axis of these DNA molecules can not only be circular or have writhe, but can be knotted or linked. So here again, some more electron microscopy images of DNA molecules. And up here, ray traced over here, this is from Shelja Pathania in my old lab, is a knot, you can see. So it's just the kind that you tie in your shoelace, but you seal the ends together afterwards. So it's a circular molecule that's knotted. So this knot has 6 crossings. Here's another 6 crossing knot over here. DNA can also become linked, as shown here, and even in more complicated ways in kinetoplast DNA over here. So this knotting and linking occurs, knotting and linking both occur during the process of DNA recombination that DeWitt will be talking about next. And DNA linking also occurs when cells with circular genomic DNA, such as E. coli, copy, the DNA becomes linked. And so just as we have a way to quantify the notion of writhe, we have a way of quantifying this notion of knotting or linking. And so if we just look at 2 examples from the previous slide, here I've ray traced out this 6 crossing knot, and we say that the linking number of this is 6. So for the topologist in the room, I'm being a little sloppy here because we're really looking at the linking number of the 2 edges of the ribbon that make up this ribbon knot. But the 6 here refers to there's no way to draw this particular knot with any fewer than 6 projections. 6 crossings in any projection. Here's a link of 2 components, ray traced from over here. This is just writhe down here, DNA supercoiling along this bit, for example. So we have the DNA molecule here with 4 crossings shown here. So this linking number of this has 4. And so what we see is that we have these 3 descriptions of the interestingness of our DNA molecule: the notion of twist, the notion of writhe, and the notion of linking. And a very beautiful theorem proved by Caloguranu and Jim White and later translated into the biological nomenclature by Vinograd is this beautiful relationship that the linking number of 2 closed curves in space is equal to the sum of the twist and the writhe. So if we have, let's just make sure we understand what that means. If we start with a linear DNA segment with the 2 backbones, the 2 sides of our ladder shown in red and blue, we can either unwind one turn, so untwist it a small amount and seal, or we can add some writhe to it and then seal. And these 2 are topologically equivalent. The twist and the writhe will sum in both cases to the linking number of 9. So this partitioning between twist and writhe is a really beautiful relationship in DNA. So now I've discussed for a few minutes about DNA and some interesting properties it has. I want to now tell you about the second word in the title, which is the word topology. And this talk, for the topologist in the room, I apologize, but this talk is aimed towards non-topologists. So this is a very famous series of images. I actually think it's easier for me to read it backwards, right to left, but you can read it forwards if you want. But the idea of topology is sometimes called rubber sheet geometry. It's looking at questions of shape and structure that are preserved under continuous deformations, under stretching, rotations, translations, but not puncturing and not sealing. And so the famous example is that a coffee cup is equivalent in a topological sense to a donut, the surface of a donut. That's contrasted, just to make it clear, that's contrasted with the notion of geometry, which comes from the Greek geo meaning earth and metri meaning measuring. And it's that second part of the word, measuring, that really distinguishes geometry from topology. And it studies questions of shape, as does topology, but also relative position of figures and the properties of space itself. But in particular, geometry requires this notion of a distance, also known as a metric. And that metric, the distance between you and me sitting in this room, can be measured just in terms of the normal distance, or in more interesting geometries, those proposed by the late Thurston, then they can have hyperbolic or other geometries as well. But they always have a notion of distance between points in a way that topologists don't. So I've explained now what topologists think topology is and what geometers think geometry is. And now I want to explain to you what biologists think topology and geometry is. And that is the phrase DNA topology, more accurately in the Newton Institute, means it's a description of both the geometry and the topology of DNA molecules and their interrelationship. And that can all be discussed in terms of these three parameters that I mentioned already: twist, writhe, and link. So here again, just to make sure we're clear, this is the linear DNA segment closed up into a circle with linking number 25. We can now unwind it by 2 right-hand turns and reclose it up, and these 2 molecules are equivalent topologically. They both have linking number 23, but the twist and the writhe are partitioned differently, here with writhe and here with twist. So DNA topology is the study of these properties: link, twist, and writhe. So I've explained what DNA topology is, but a maybe more pressing question is, why would you care about it? And I want to give 2 short examples for what reasons people other than basic scientists may be interested. So we're going to look at DNA topology and chemotherapy. So this is the tree Camptotheca acuminata. It's found in China. It's also called the cancer tree or the happy tree. And there's a compound from it, camptothecin, like this, whose several analogs have been used in chemotherapeutic drugs. So here's a picture. Let's see what this camptothecin and its analogs do. Here's a picture of the same camptothecin molecule bound to a protein that I haven't told you about, a mystery protein called topoisomerase 1. And both of those are bound to a DNA molecule, a segment of DNA. So camptothecin works by binding to a mystery protein, and that mystery protein is called topoisomerase 1. So what does topoisomerase 1 do? How does camptothecin work as a chemotherapeutic drug? It works by harnessing this formula that we just learned about, link equals twist plus writhe. So here, if we have duplex DNA, the topoisomerase 1, this is a low resolution image, will cut one duplex here, pass the other through, and reseal. There's 2 different types of topoisomerases, but for now I'll just discuss this type of topoisomerase 1, but I'll just discuss this type. So if we look at it in a circular molecule, then we see that these topoisomerases regulate DNA writhe by harnessing this formula that we've just learned about, link equals twist plus writhe. So here we have a circular double-stranded DNA. We remove a few turns of the double helix, as I just showed in the preceding slide, by cutting, pass through, resealing. This is of one backbone through the other. So these aren't double-stranded breaks. These are single-stranded breaks. So we reduce the twist, and that in turn compensates by adding or reducing the amount of writhe in the molecule. So topoisomerase 1, this target for camptothecin and the analogs to it for chemotherapeutic drugs work by harnessing this formula, link equals twist plus writhe, to affect a change in twist that then leads to a change in writhe and DNA supercoiling, which is the native state. DNA is natively supercoiled in the cells. And camptothecin works by stabilizing the complex after the cleavage and before ligation, so stabilizing this intermediate. So this is one example where DNA topology, or DNA geometry, is important medically. The second type I want to talk about is a drug. This is Podophyllum peletatum, which is also known as American mandrake or May apple or hog apple. Anyway, many names. It's from the American Northeast in forest. And a derivative of that is etoposide. Again, toposide should make you intrigued. So here's a structure of etoposide bound to DNA and a molecule, a mysterious protein, shown here, called topoisomerase 2, shown here. This is from Science last year, from Wu et al. a beautiful crystal structure. So etoposide works similar to camptothecin by trapping an intermediate complex. And the complex now is DNA, the drug, etoposide, and a mystery protein. And now the mystery protein is topoisomerase II. And so the question may be, what does topoisomerase II do? So it regulates DNA knotting and linking. And its dominant reactions, although it does others, is to take double-stranded DNA molecules that are either knotted or linked and unknot and unlink these molecules. Okay. And so there's one other arena where DNA topology comes into play, and that's in the world of antibiotics. And so topoisomerase II is also a target for the quinoline family of antibiotics. So here's a structure of all the quinoline family here. If there's a fluorine here, this is the fluoroquinolines. And over here is a crystal structure shown with a DNA molecule, 2 drugs from the quinoline family, in this case ciprofloxacin, bound to a type 2 topoisomerase called DNA gyrase. So these are examples. In fact, these antibiotics are the most active and broad spectrum antibiotics currently in clinical use. So this is just a brief introduction, but I hope I've been able to at least inform you a little that DNA has non-trivial geometry, which we recognize as rive, and that rive is typically good. DNA is typically supercoiled, has negative rive in the cell. It's maintained and controlled by this protein, topoisomerase I, which harnesses the formula link equals twist plus rive to do a change in twist, which leads to a change in rive, and that topoisomerase I is an important chemotherapeutic target. Also, DNA has non-trivial topology. That is to say, the axis of the DNA molecule forms both knots and links. And unfortunately, that topology, that link, is knots and links are actually typically bad. So they're systematically removed by topoisomerase II, and if that process isn't effective, so these topoisomerase II poisons can be important, both chemotherapeutic drugs and antibiotic drugs, to trap the intermediate complex. Now I'll turn over to DeWitt, who will tell us a little more about the process by which these DNA knots form.
We'll defer questions until DeWitt has his say.
I do have a talk here someplace. Thanks. So thanks, Dorothy. I'll start with a few statements about mathematics. Since this is a sales pitch for mathematics, here's what Galileo said. You know, the book of nature is written in the language of mathematics. And here's what Darwin said, being an honest Cambridge man. He said, I need to learn some more mathematics. Yes, there's a guy with really good taste. But in the 20th century, what were the big scientific revolutions? Well, to me, it was the computer, and it was, of course, DNA here in Cambridge at the Cavendish lab. And in molecular biology, as you heard from John Toland's letter, the human genome has been sequenced. You have huge amounts of data acquired, microarray chips, so on, just a flood of data. We also have computational ability, which is huge and growing, in order to understand what this data is telling us. So you can think of mathematics as a way to take sort of sparse data points and maybe put threads of understanding between and among the points and weave these threads together to give you perhaps a fabric of understanding. On the computation side, you build algorithms to breathe life into the mathematical theory and to run simulations to enable understanding of these massive biological data sets. So here's DNA replication. Dorothy told you something about it. And as you see, the fact that it's twisted up, and this is replication, means that the backbone strands form templates for the daughter strands. Since the backbone strands are interwound after replication, it means that the daughter strands are themselves going to be topologically entangled. So the question is, how does the cell fix this? The cell fixes this through topoisomerase, which, Dorothy showed you some much more This is to a mathematician, what topoisomerase does. The DNA is represented by lines, forget the double helix, and the topoisomerase is the blob. And what topoisomerase does is it attaches to DNA, breaks one DNA molecule apart, holds the ends, passes the other one through, reseals the first one, and lets it go, thereby, as if by magic, passing the DNA through itself. And using this mechanism, the cell can solve all sorts of possible entanglements, like the one you see in replication. And then there's another one that I'll talk a little bit about, site-specific recombination, where an enzyme known as recombinase attaches to two strands of DNA, breaks one and the other, and then glues the red to the yellow, changing the genotype. So this is the way that, for example, that viruses inject their DNA into the genome of the host and then extract it later. And it's going to be very important for the talk that Lynn's going to give, because it's a recombination that's the magic step in producing DNA minicircles. So here's the problem. Here's your enzyme. There's the DNA. This is an electron micrograph. And what you see is the enzyme is like my fist and the DNA is sticking out on both sides. You'd like to tease apart what's happening. This is the enzyme that's doing something really interesting to the DNA. So you'd like to use mathematics to get at what's happening in the box, to kind of pull back the curtain and look at the secrets. So the method of topological enzymology was developed, whereby you take small circular DNA molecules, plasmids, 5 to 10,000 base pairs. You use them as reporters to tell you about the enzyme. And this is in vitro experiment, so you have purified protein, the enzyme, you put it in a test tube, you take a family of DNA supercoiled molecules like this, you feed them to the DNA, it slices and dices. Out the other end come changed DNA. Changes the amount of supercoiling. Here there are 2, here there's 1. These are unknotted. You could untwist them to open circles. Here's a knotted DNA molecule. These molecules start out being unlinked. They could end up being linked. You can observe these products. You can control what's happening to your input. And by observing the change in geometry and or topology, the idea is to use mathematics to deduce enzyme binding and mechanism. So you'd like to figure out how the enzyme binds here. Are there some supercoils trapped? What happens when the, say, recombination takes place? So that's the kind of game we want to play. And so you react circular DNA, you do electrophoresis to fractionate the products, you do electron microscopy, and finally, you use some mathematics. Yes. So these first 3 steps take postdoc years. This last step, well, you do it in the Eagle, just like Crick and Watson did. You go to the Eagle, get a beer, get a napkin, do some math. That's the idea. So, of course, that's why I'm a mathematician, because I can't do this other stuff. It's too hard. This stuff, yes, okay. So that's what a gel looks like. I won't go into the details because I don't really understand it myself. It's pretty difficult sort of scientifically to understand it, but you get these nice pictures. Here's Rec-A that Dorothy talked about. So it's a protein that mediates general recombination in E. coli. So you can use it to coat DNA molecules, so you can see, if you take an electron micrograph, it gives you the resolution to see the crossings. Like this is naked DNA. It's hard to tell which is over and under in this crossing. But in this fattened version, you can see that this one's over and that one's under. And so you can do experiments. Here's an experiment done on topoisomerase. You feed in unknotted DNA, and out come all sorts of knots. Here's some more of them. Just, so the entire family of knots up to, say, 9 crossings. One of the important things that you can get from experiments like this is to learn that the knots migrate through the DNA according to how much knottedness they have. So this is a 3-crossing knot. It's a little slower. It's faster than the 4-crossing, the 5-crossing. You have this linear relationship between knots and their gel velocity. So this turns out you can sort of, without seeing the pictures, you can figure out which knots you've got by looking at the gel. So that's very important. So here's a nice picture of a DNA trefoil knot. I mean, this is the picture that got me into the business. I have a degree in pure mathematics from Cambridge, of all places. And, I got promotion and tenure doing knot theory. But when I saw this picture, I knew it was full employment for mathematicians right there. Because, look, this is coming out in the lab. This is a wonderful picture. So that's one of these topoisomerase knots. Here's a knot produced by site-specific recombination, bacteriophage lambda. Now, Lynn, are you using lambda in your You're not getting this, you're getting little circles. But so this is what you get. And so let me talk about site-specific recombination. So here I've got circular DNA, and I've got some short bits of DNA. They say 10 to 15 base pairs long, and these enzymes, the recombinases, can find these sites where the active region is, can bind to them, and then break them apart, gluing the red bottom to the black tip and the black bottom to the red tip, doing this crossover event. You want to study what's happening, so the way you do this is you can engineer, by cloning techniques, put both sites on the same artificial DNA plasmid circle for your experiments. You can put them so that they're head-to-head in orientation. They have an orientation because the word that you read in A, C, T, and G, the base pairing, is non-palindromic. So without loss of generality, it has a local orientation associated to it. So here's sort of inverted repeats, and here's direct repeats. And I believe this is the kind that Lynn's going to do. It's directly repeated sites. And when you do, here's the enzyme bound to DNA. The difference between direct and inverted repeats is in direct repeats, here I have a supercoiled substrate with directly repeated sites. You take this arrow and it lines up with that arrow. You do recombination once, this single circle becomes a pair of circles. Here's the protein bound to it. Here's the result of one round of recombination. If you were to start with a pair of circles and do a round of recombination, it'd take you back to a single circle. With inverted repeats, you always stay with circles. But for Lynn's talk, this is what's happening in her experiment. In her company, yes, she's going to sell this stuff. Okay, so this is the resolvase synaptic complex. So resolvase is a transposon from E. coli. And the idea is to do the mathematics of so-called tangles. The idea is to look at little small regions of space. The protein itself forms like a little blob, and you have two strands of DNA inside the blob, forming sort of a geometric configuration of two strands of DNA bound to a protein. So mathematically, that's known as a tangle, and it's possible to do the mathematics of tangles to compute structure from the electron micrographs. So here's the idea. You have a protein that's bound to the DNA, and inside the DNA, you might have some supercoils that are bound in a sort of a geometric footprint to the protein. These are where the sites are. This is where the recombination takes place, and this is everything else that's in the picture. The idea is to think of these as unknowns and to compute them from the pictures themselves. And the pictures look like this. You start with this, get one round, you get a catenane like this, then you do 2 rounds, and you get the figure 8 knot, and so on. So it's possible to prove theorems. So if you like, this is a mathematical proof of structure and mechanism. Of course, when you try to tell this to a biologist, is it really a proof? Well, for a biologist, the proof is you make a hypothesis about what's going to happen, you do the experiment, and either it comes out the way you said it's going to come out, or it doesn't. But I mean, you can actually prove a theorem here, and after a number of years, I was able to finally sell Nick Cosarelli, who did all this work in his lab, that if he had known that all this theory was possible, it would have saved him at least one postdoc year. I got him to say this in public one time. Anyway, using these theory, you can prove that if you do recombination often enough, you'll get this knot, which in fact was observed. What's the utility of the mathematics? Well, it gives you a precise mathematical language. It allows hypothesis testing. It calculates all the alternative pathways that you can do to get to these products. And it can be used with incomplete experimental evidence, like no electron micrographs. Here's the other bit of good news. We wrote this paper for JMB. It was a nice cover article. The problem was there were 5 authors, and the number of people in the world that read it, maybe 10. We had 5 of which were the authors. But, at least you see some tangles on the cover. Yes, so it's possible to do mathematics to kind of get an idea about molecular action on DNA. So with that, I'm going to turn it over to the main act, Lynn Cedric.
Very good. Well, thank you, Jim. I should have said in my introduction that DeWitt did his PhD at Pembroke College, Cambridge, here in the 1960s. 67, was it? Just
Good old days, yeah.
Just when John Conway was introducing the concept of tangles.
Tangles, yeah, that's his idea. Yeah.
Around the same time. So thank you. Dorothy and DeWitt. And now it's my